Hello, MIP611. My name is Selena Nieto, and I am going to be doing my second local expert project on stem cell culture. So what exactly are stem cells? Stem cells are cells that can develop into any type of cell line during the early development of the organism. These stem cells can be broken down into two main categories: pluripotent, which are like the embryonic stem cells, and non-pluripotent, or adult stem cells. There has been research where these stem cells collected by a donor or from the individual treated such cured diseases such as cancer, which we will look more closely at later in this presentation. Stem cell culture connects to this class with the last lesson, lesson 5.1, where we talked about cell culture. However, there are extra steps. These stem cells need to be stem cells the whole time during the process of culturing. They cannot be differentiated into different cell lines. Firstly, these cells are grown on plates where they usually feed on mouse embryonic fibroblast or human foreskin fibroblast. Along with this, normal cells that are assigned a function such as a skin cell can be induced back to pluripotent stem cells by forcing the expression of genes that are normally highly expressed in these stem cells. So either you have your stem cells and you continue culturing them and passing them for as many times as you can, keeping them as stem cells, or you can get a cell line such as a skin cell line and induce the cells by expressing genes that are normally found in pluripotent stem cells to induce it into its stem cell state. So now that we know what stem cells are and how to culture them, what are the advantages and disadvantages of using stem cells for culture? Some advantages include low to no risk of donor rejection. And this is extremely important because a lot of, for example, blood transfusions or organ transfusions that are done can lead to the donor rejecting it by causing an over-exaggerated immune response. And there are actually also clinical studies where scientists use stem cells from a patient and do in vitro experiments where the stem cells are brought back to the same patient as a treatment for a particular disease. And this will ultimately eliminate any chance of a donor reaction because the stem cells that are used for this experiment are the stem cells from that actual patient. And another advantage, embryonic stem cells can help us learn about early development. And also the research completed to induce a cell line, such as the skin cell line back to a stem cell line, can potentially be used to quote unquote reprogram damaged cell tissues in the future. And again, however, with any advantage comes many disadvantages. And with stem cell research, this usually revolves around major ethical concerns. Some of those include induced pluripotent cells, when under certain conditions can actually become embryos. Also, to retrieve embryonic stem cells, the embryo is usually killed, and therefore it is not an easy task to gain consent from the mother for these stem cells, since it will ultimately harm or kill the embryo. And although adult stem cells are much safer to obtain, as in they don't really cause any harm, they are not as beneficial as embryonic stem cells, since embryonic stem cells are able to differentiate into multiple different types of cell lines. Adult stem cells are kind of lacking that. So now that we went over what stem cell culture are, I found 3 figures from 3 different journal articles that I'm going to show to you here. This one is figure 1, and it shows stem cells at different times and under different conditions with different stains. And I thought it was a pretty nice picture. I think it was pretty cool. Again, here with the next paper, it is stem cells underneath different conditions after a certain amount of time. And as you can see, they were growing and flourishing on day 2, but on day 7, not so much. And it's crazy to say that like it just looks just like normal like cell culture pictures that we looked at in class. So yeah, thought that was also a cool picture to see. And here is the last one, which is my favorite, save the best for last. This is actually showing cells after radiation after a certain amount of time. And the cells that are in red are actually the ones that have died. And as you can see in figure B, the more time that went on after radiation exposure, the more cell death that has occurred. And I thought that was pretty interesting to actually like see it in a picture. But anyways, that's all I have for you guys today. Thank you for watching my second local expert video, and I hope you guys have a great day and a great winter break. Bye!